Hi, everyone. Welcome to What's New in MPMI. While people are coming into the room, I just want to, while we're waiting for everybody to log on, why don't you enter where you are into the chat? It's always fun to see where people are coming from. We're a really international society of MPMI, and it's great to see where people are coming from. So let's see. Someone says they can't hear me. Sonali, could you hear me?
Okay. Yes, I can hear you.
Okay, not sure what's going on there. Okay, we have Sridhar Raghunathan coming from Chennai, India. We have Bhavana Gimir from Tennessee State, David Mackey from Ohio, Dong Wang from Amherst, Massachusetts, Jessica, let's see, Marcel Boucher from Cologne, Germany, Christina Arthur from Wisconsin. Hi, Christina. Ankita Alexander from Boston. Let's see, Edmund Bridge from Norwich. And Abel Wonky from Cambridge, Fernanda de Carvalho Nibel, hope I got that one, from Toulouse, France, Stefano Leite from Italy via the Netherlands, and lots of people coming on, Jessica from Puerto Rico. So, okay, now that we've got everybody joining, I just want to welcome you to What's New in MPMI, which is the virtual seminar series of the MPMI journal. And we've been doing this for over a year now to showcase the works of various authors and to showcase some of the papers in our journal and give you a chance to meet the author and interact and ask questions about the paper. So before we start, a few little announcements. MPMI is looking for new assistant features editors, and that's been a really terrific experience with our first three. And in fact, one of them, Morgan Carter, is going to be live tweeting this seminar. So that was one of the things that she has added. And Ashley's going to put the information for applying to become an assistant features editor into the chat, so you can take a look there. Sonali Roy, who is our speaker today, was an assistant features editor for Plant Cell, and actually that's how I first got to know her. So it's a really good opportunity. I also wanted to let you know that MPMI is going to have our next focus issue is on the endomembrane system and its role in plant microbe interactions. And we have a page for that Ashley will type in as well. We hope to be collecting submissions over the summer up till September. And finally, one last announcement is that in order to increase accessibility and promote inclusion, we are having our transcripts edited so that it's easier for people, anybody who's hard of hearing, to follow, and also people who are, for whom English is not a first language, may find the transcript an easier way to engage with this content. So we need somebody, if you would like to volunteer some service to your society and to help make this a more inclusive seminar, if somebody would like to volunteer to edit the transcript that Zoom makes. So Zoom does a terrific job with the transcript until it hits scientific words and personal names, and that's where you come in. So if this is something you'd like to do, please contact Ashley via the chat and we'll get in touch with you. Okay. All right, now moving on to our main event, I'm delighted to introduce Sonali Roy, who is today's speaker. And as I said, I first met her via her work at the Plant Cell. And I chose this paper because it's such an interesting connection between two very important nutritional symbioses, which I find really fascinating, rhizobium legume interaction and mycorrhizae. So I think that it provides some nice insight into what's going on. And Ashley has posted the link in the chat. So remember, if you want to go deeper, you want to look at the details, go ahead to the paper itself and you'll find a lot of it there. And also you'll find contact information for Sonali Roy, who is the senior author on this. Okay, well, I'm going to turn it over to you, Sonali. Thank you so much for coming to speak.
Yeah, thank you, Jean, for having me. And as an assistant features editor, I would highly recommend, for Plant Cell, I would highly recommend the program also, which MPMI has. It gives a lot of exposure to how science editing is done. And just to be involved with the entire process was a great experience. So I highly recommend it. Okay, so I will get back to my talk and start sharing. Okay, thank you, everybody. Good morning, good afternoon, good evening, and thank you to everybody who's joining me today for my talk. My name is Sonali Roy. I'm an assistant professor at Tennessee State University in Nashville, Tennessee. And today I'm going to be presenting research that I conducted when I was a PhD student in Jeremy Murray's lab at the John Innes Center, and which was published last year in MPMI. And as the title says, the topic of my talk today will be 3 common symbiotic ABC subfamily B transporters in Medicago truncatula that are regulated by a NIN-independent branch of the symbiosis signaling pathway. So before I begin, I wanted to give an overview of my career path. I started as an undergraduate student. I studied for a BSc in botany at the University of Delhi. From there, I moved on to work towards a master's in plant molecular biology and biotechnology at a research institute in Calcutta called Bose Institute. Then I moved to Jeremy Murray's lab for a PhD at the John Innes Center in Norwich, UK. From there, I went to Professor Michael Udvardy's lab, where I was a postdoctoral fellow for 5 years at the Nobel Research Institute. And as of January 2021, I'm starting my own lab as an assistant professor at the Tennessee State University. So the research that I'm going to be presenting today, I started in 2011 with Jeremy. And at that time, Jeremy's lab was interested in understanding the molecular mechanisms that underlie legume interactions with beneficial microbes that included bacterial symbionts called rhizobia and fungal symbionts, which lead to development of arbuscules. So, and this image aptly summarizes the research that I was involved in. So on the left side, you can see an image of a root with root hairs responding to rhizobia. So the root hairs are curling in response to the bacteria. And on the right-hand side, you can see cells stained in blue, which host the arbuscule. So what is root nodule symbiosis? Root nodule symbiosis is an association between legumes. It is restricted to legumes and a few other land plants. And it can help plants acquire nitrogen in exchange for carbon. And it results in the formation of an organ called nodule. And that is what the symbiosis derives its name from. It's called the root nodule symbiosis because of that. In comparison, the arbuscular mycorrhizal symbiosis is an association that can be found in more than 70% of land plants. And these plants associate with some zygomycete fungi, and which help them acquire phosphorus and other nutrients in exchange for carbon. So AM symbiosis results in the formation of a specialized nutrient exchange structure called the arbuscule, after which this association is named. So throughout my talk, I'm going to switch between the root nodule symbiosis or the RN symbiosis and the arbuscular mycorrhizal symbiosis or the AM symbiosis. And to make it easier for you to follow, I will have these 2 thumbnail images. So if you see the nodule to your left, if you see the nodule to your left, I'm talking about the root nodule symbiosis. And if you see the arbuscule, it is, sorry, I'm trying to, I'm talking about the AM symbiosis. Okay, so both AM symbiosis and root nodule symbiosis have many similarities and parallels in how they are initiated. So the plant usually, when it experiences, the legume experiences, it feels that there's not enough nitrogen, it secretes a signaling molecule, which is often thought to be flavonoids. And when these are perceived by rhizobia, they in turn secrete chemoattractants or signaling molecules called nod factors. Once the plant perceives these nod factors, it allows the rhizobial bacteria to attach. The root hairs curl around them, form a microcolony, and then the bacteria start traversing down plant membrane encased infection threads towards the cortex. So a successful infection is marked by the formation of a simultaneous formation of a nodule right underneath this infection thread. And in Medicago truncatula, the model plant that I study, an indeterminate nodule is formed. So an indeterminate nodule has 4 distinct zones, which is the zone 1 meristematic, zone 2 the infection zone, zone 3 is the nitrogen fixation zone, and there is a senescence zone. So in comparison, the mycorrhizal symbiosis also initiates when the plant secretes a signaling molecule. In this case, it is the hormone strigolactone, which is then perceived by the fungal spores in the soil, and it causes these fungal spores to germinate and form hyphae. And when these hyphae first touch the root surface, it leads to the form of a specialized aprosoria called the hyphopodium. So just beneath these hyphopodium, as the plant gets ready to guide and invite the hyphae in, it forms a pre-penetration apparatus, which is very similar to the infection thread. And as just what happens below the infection thread, and as the hyphae enter the root cortex, it starts branching and dividing, finally forming the arbuscule, which is the main site of nutrient exchange for the fungus. So at least 12 genes are known to be shared between the root nodule and AM symbiosis, based on all the collective efforts of our community over the past 20 years. We know that downstream of the receptor, which is specific to the MIC factor or the nod factor, there is a symbiotic receptor kinase, which is the SYMRK in lotus or the DOMI2 in Medicago. There are 2 ion channels, castor and pollux, or DOMI1 in Medicago, 3 cyclic nucleotide gated channels, a calcium calmodulin kinase, also known as DOMI3, Cyclops and its 2 Medicago orthologs, interacting partner of DOMI3 and DOMI3-like, Vaporin, lumpy infections, and 2 GRAS domain transcription factors, NSP1 and NSP2. So we have been working as a community trying to study both these symbioses, and research has been ongoing for quite a while. But why should we study these symbiotic interactions, right? So I think right now is a very exciting time to be a part of symbiosis research. And this is because groups world over are trying to make non-nodulating plants nodulate, so that they will not be able, so they can fix their own nitrogen, and they will be independent of any added nitrogenous fertilizer requirements. So of the 3 approaches that are being taken to achieve this goal, well, the legume genetics contributes to the second one, in which we can try and make crops better hosts for nitrogen fixing bacteria. And to achieve this, we have to engineer a nodulation in nodulating crops. We have to think about the early signaling for the plants to be able to perceive signaling molecules. Then we have to think about rhizobial infection. And the third is organogenesis itself. So there is growing evidence from Giles Oldroyd and Jean-Michel Anne's labs, which show that organogenesis is possible in non-nodulating plants if you can just tweak, if you can modify the cytokinin signaling biosynthesis pathway. But engineering infection competency still remains a challenge. So that's why it is really, really important. It's crucial to study rhizobial infection processes to engineer functional symbiosis. So that's exactly what we were trying to do in Jeremy's lab. So when I joined the lab in 2011, there was a postdoc already, and he had set up this really cool method to try and isolate root hairs, and which I really enjoyed learning. And so what we did, we used to grow these plants for plants on plates, infected them with rhizobia 3 days or 5 days post-inoculation. We took these seedlings and we had around 150 per biological replicate. We dunked them into liquid nitrogen in a baking tray, and we used a paintbrush to brush off these root hairs. And to show that this process was effective, if you look at the images of the root hairs, before we brushed them off, you could see, if you stain the roots, you could see a lot of infections and infection threads. But after which, if you look at the denuded roots, you can see that only the root hairs were collected. So these were purified root hairs, which we then had which we could extract RNA from and send off for transcriptomic studies. Specifically, we used RNA sequencing and the microarray chips that were available in Medicago. So at that time we were doing this transcriptomics, there was another really cool study completed in LIPM Toulouse, and it was done by Bryce Roux et al. And what they did was they used the laser capture microdissection technology, and they isolated just the zones of the nodule. So the meristem zone, the zone 2 infection zone, interzone, zone 3, and the senescence zone. And they got enough tissue then to perform a transcriptomic analysis again. And what we then did was compare these transcriptomes that they published with our own in-house root hair transcriptome. So what we found was there was a core set of infection genes that were enriched both in infected root hair cells, and these were also expressed in zone 2 of the nodule, which is expected kind of, because the zone 2 is where the infection threads are within the nodule itself. Okay, so coming back to the root hair transcriptome, we looked at the top 1% of the genes that were induced, and these included 3 ABC transporters. And their expression was so striking. So we looked at their expression in the gene expression atlas, which doesn't exist anymore. But the expression was so striking, it was not expressed under any condition, except if the plants were infected with AM fungi, or in root hairs or nodules when infected with rhizobia. So with that, that's how we named these genes. We called them ABCB transporters in mycorrhization and nodulation. And so AMN stands for that. So what are ABC transporters? ABC transporters are ATP binding cassette transporters, which are ubiquitously present across all living organisms, from microbes to humans. And they use ATP hydrolysis to transport a variety of substrates. They could be transporting anything. And they have two, based on the number of nucleotide binding domains and the number of transmembrane domains, they can be classified as full and half transporters. So full transporters have 2 nucleotide binding domains and 2 transmembrane domains, and half transporters have exactly half of that. And they have to dimerize for them to be functional. So in plants, there are 8 families or subfamilies of ABC transporters from A to H, and ABCG is the largest subfamily of transporters within plants, even within Medicago truncatula. ABCG, sorry, and ABCB is the second largest subfamily of ABC transporters. So when I started in 2011, the genome sequence had just been made available. And so what I did was I tried and defined the entire complement of ABC transporters in Medicago. And for that, I took the ABC transporters known in Arabidopsis and did multiple blast searches within the genome. And this I verified with the latest genome version 5 as well. And we found that there were a total of 186 ABC transporters. So it's a very large family of ABC transporters in Medicago, and of which there are 107 full ABC transporters. And this phylogenetic tree shows the classification and the interrelations between these subfamilies. So in red are the ABCB transporters. And as you can see, AMN 1, 2, 3 belong to this subfamily. And next, what I did was I looked at because we found 3 ABC transporters which were responding to symbionts, I tried to see what other ABC transporters were also responding, just to be very thorough that I had the targets that I needed to follow up. And so of the genes that were responsive to AM fungi, there was a total of 42 out of 186 ABC transporters that were transcriptionally upregulated by mycorrhiza. And I found that there were several other which were also responding in nodules, but there were only 3, the AMN 1, 2, 3, which were most strongly induced in root hairs. Root hairs, in addition to being MYC responsive. So at that time, because they were getting induced under these symbiotic conditions, we thought, can we find more information about their function based on just orthology to better studied plants, let's say in Arabidopsis or other plant species? So we wanted to ask, are there also orthologs in other plant species? And for this, I selected 26 plant species which have been sequenced, and their genome is available on NCBI. And this included several determinate and indeterminate nodules, several plants which could associate with AM fungi, but not with rhizobia, ectomycorrhizal species and non-mycorrhizal species belonging to both the Brassicaceae type and the Proteaceae type families. And then I looked based on homology again, or orthology, are there genes which are present, are there AMN orthologs present in these species? And this is a phylogenetic tree which shows all the ABCB transporters in Medicago truncatula and orthologs from the plant species that I could find for all AMN 1, 2, and 3. And what it clearly shows is that the closest orthologs of the Arabidopsis ABCB15 and the Nilambo ABCB1 and 2 did not cluster together with the AMN 1, 2, or 3. And only plant species which could either form the AM symbiosis or even the ectomycorrhizal symbiosis, in fact, had encoded these AMN transporters. So during the review process with MPMI, Jean Harris asked me an interesting question, and I learned something new here. It was just to, can we conduct a statistical test to show that this association is not random? How do we know that if we just don't take any random 26 species, we will not find the AMNs in some other plants? So we conducted something called a Fisher's exact test, and we found that the presence of AMN 1, 2, 3 and the association of these genes only in plants that could form symbiosis was highly significant, and these were only present in symbiotic organisms. Okay. So what is the role of these ABC transporters during AM on root nodule symbiosis? We know that ABC transporters are involved during multiple stages of symbiosis. Some of the best studied to date are the STAR1 and STAR2 half transporters, which are involved in mycorrhizal establishment and the formation of the arbuscule. There is also the strigolactone transporter, which is ABCG59, which has an impact on the symbiosis establishment. And another ABCG, which was recently described to be transporting cytokinins, which is required for both early and late stage nodulation. There is only one ABCB which had been studied during nodulation, and that is the Lotus ABCB1, which was thought to be an auxin efflux transporter, and it was expressed only in only in cells of the nodules which did not host the rhizobia, uninfected cells of the nodule. So what do we know about ABCB transporters from other plant systems? We know that in Arabidopsis, there are 4 ABCBs which have been studied. So ABCB 19, 14, 15, and 4. And they tend to export non-directionally the phytohormone auxin. And this is they are expressed at the root hair tips also, and they are involved in the shootward movement of auxin. So to understand what the AMNs were doing during nodulation and mycorrhization, we wanted to find out what are the temporal and spatial expression of the AMNs. And so I started by combining both qPCR and promoted GUS studies to try to understand that. And I could confirm with qPCR what we saw in the transcriptomics data. We saw that in root hair cells, AMN 1, 2, 3 were strongly induced in comparison to plants, root hairs, which were infected by bacteria that could not produce nod factor. And when I cloned the promoters, I could see the same thing, that these AMN 1, 2, and 3 all were expressed in root hairs, which contain infection structures, be it microcolony or infection threads. So this I followed up with qPCR from excised nodules at different stages of nodulation, so 4 DPI, 10, 14, 21, and 28. And I saw that all three of these were most strongly induced when in primordia, or when the nodules were just emerging, and there was a lot of meristematic tissue, and then it reduced later on. So this expression pattern is very common for genes that are expressed have localized expression within the nodule. So the AMNs expressed in zone 1 and the infection zone as well, which we could predict from comparing to the Roux et al. Study. During mycorrhization, what I did was I infected these plants and harvested the root tissue, scored them for colonization percentage, and at the same time took other plants, 3 per replicate and 3 biological replicates, and isolated RNA from them. So again, what I saw was with increasing colonization, the expression of these 3 AMNs also increased. But if you move them to soil which contains sufficient nutrients, although there was still degenerating arbuscules, the expression of the AMNs dramatically reduced or was even abolished. Spatial expression patterns showed that these AMNs were induced in root cells which contained arbuscules. So this is in red are the fungi stained, and in blue is the promoted GUS, which was associated just exactly with infection structures. So what does this suggest? This suggests that AMNs, which are expressed in cells containing both infection threads or arbuscules, where there are a lot of membrane generation, de novo membrane synthesis, AMNs are expressed in those cells, and they probably then mediate transport of something of a common substrate. And that is the hypothesis we worked with. But if they are expressed during such different symbiosis, let's say, how is their expression being regulated so that it is more specific to each of these symbioses? To do that, we looked at the transcription factor NIN, which is a central regulator of nodulation. It's called nodule inception, and the mutants do not form nodules. And so when so Cheng-Wu Liu, what he did was isolated infected root hairs from mutants, including NIN, ERN1, and NFYA1. And what we found was that 54% of the rhizobia-induced genes in root hairs were also dependent on the transcription factor NIN. And in parallel, if we looked at the mutant required for arbuscular mycorrhization 1, RAM1, 59% of AM-induced genes were dependent on this GRAS domain transcription factor. So with that, I took 2 approaches then to try and see if these genes, try and understand how they're regulated by the symbiotic signaling pathway. And I took 2 approaches, one in which I used purified nod factors from Cynorhizobium meliloti, which is the bacterial partner for Medicago, and as well as infecting them with rhizobia and harvesting the root tissue 7 days post-inoculation. So with this, the mutants that I used were NFP, which is the nod factor perception mutant, DOMI3, which is the CCAM-K mutant, RAM1, NSP1, NSP2. These are 3 GRAS domain transcription factors, and NIN. And what I found was that in NIN, the expression of all these 3 AMNs was highly induced compared to even to the wild type. And this was more pronounced if you used just rhizobia. And 7 days post-inoculation, we could see that, as expected, the expression of these were dependent on NFP, which is the NOT factor receptor, and the CCAMK, but it was hyper-induced in the NIN mutant background, suggesting and we concluded from it that expression of AMNs are independent of NIN. When we looked during micronization, and here I looked at also the expression of PT4, which is the phosphate transporter 4, which is which gives you an estimate, which is a proxy for the colonization percentage, and it shows that these mutants did get colonized to a similar level, let's say for wild type and NFP1 and SP1. And what I found was that the expression of the AMNs was dependent, again, on DOMI3 during micronization as well, and on RAM1. And this we could confirm later with independent, this was confirmed later by independent studies, which did transcriptomics, and we found that RAM1, AMN 1, 2, 3 expression was reduced in RAM1. Okay, so now we know the expression of these genes, we know how they are regulated, that they're probably independent, they are independent of NIN and dependent on RAM1. What about their function? So to characterize them, when I started in 2011, we did not yet have CRISPR mutagenesis. So we had the TNT1 resource at the Nobel Research Institute. And what I did was I isolated 2 mutants, AMN1 for AMN1, 1 for AMN2, and 2 for AMN3. And I back-crossed them. And these lines looked super healthy. They were doing well. They had healthy roots and shoots, and they had no non-symbiotic phenotypes, which was what I would have expected. And this is just a gel image showing that the TNT1 mutant was present within the gene, within the exons that we targeted. When we looked at the expression, when we tried to quantify the expression using qPCR, AMN2 and AMN3, we could clearly see there was a 50 to 80% reduction in transcript level. But AMN1, surprisingly, it had the transcript, it was as if it was getting more induced in the mutant backgrounds. And how we explain this was often if a plant does not get enough of a gene that it requires, it tries to push the transcript production, or and that is what we think we are seeing with AMN1, that the plant is trying to make more of the transcript when the functional protein is absent. So when what I did was I back-crossed them, then I made double mutants, and then I also made a triple mutant. And they were available in 2014. By the end of my PhD, I had them ready. And when I phenotyped them, I saw that the AMN 1, 2, and 3 did not have any nodulation defect at the 3 weeks time point. And during mycorrhizal formation as well, the single mutants had, compared to wild type, they made exactly the same. They had total percentage colonization was similar. They had a similar number of arbuscules and vesicles. And this was also the same for the double mutants. So the colonization percentage was not different from wild type. And finally, when I looked at the triple mutant, and these were studies conducted by different people in different countries, in fact, in Jeremy's lab in China now, and at the Janenis. And what we found was that the percentage colonization was not different between wild type and the triple mutant, but it is possible. The first experiment, one of the experiments showed us that if the colonization percentage was weaker, there was maybe a slight reduction in colonization, but this is not significant, and it was not very reproducible. So we cannot conclude anything from that. When we looked at nodule formation, however, what I kept seeing was that there was a slight increase in the number of nodules. And when I did this experiment many times in the UK, in America, in the US, and also my colleagues in China tried to repeat it, we found that exactly in 50% of the experiments, there was a hypernodulation, and 50% there was not any effect. And so this might be I think it might be a conditional phenotype that we are seeing, depending on on the timing of the inoculation, but we need several more alleles of the triple mutant to be able to be confident of the phenotype itself. So with that, I completed that study, and what we can conclude from this work was that we had 3 new ABC transporters which are induced during both AM and RN symbiosis. AMNs are encoded only in species associating with AM fungi or rhizobia, or as forming symbiotic associations, and this expression of AMNs during nodulation is not dependent on the transcription factor NIN. In terms of what we can do next, of course, we want to know, are there other ABCB transporters that are compensating for AMN activity? And is that why we are not seeing a very strong phenotype? And with CRISPR, we are able to do that now. And also the other burning question, of course, is what do these AMNs transport? And there is some evidence from other groups which we collaborate with, and hopefully that will be published soon too. So with that, I would like to stop and thank everybody who helped me when I was doing my PhD. So Jeremy Murray was my advisor, and Chung-Woo and Andrew Breakspear, they together were the postdocs who did a lot of the initial transcriptomic studies. Kirsty, Donna, and Anil helped me verify all the phenotypes that I saw. And Yang Su helped me verify the phenotype as well in Ping Zhu's lab, and Michael, whose lab I was in recently. And Giles Oldroyd and Alan Downey's lab together. When I did my PhD there, it was an incredible environment to work at the John Innes, because we had 3 leaders of the symbiosis field, and I really enjoyed it. So thank you for listening, and I'm happy to take any questions.
Thank you, Sonali. Please join me in thanking her for a really interesting and informative talk. So if you are interested, if you have any questions, please type them into the Q&A and we will take turns reading them, but not into the chat. So I'm going to start off with a simple one while people are typing in, Sonali, and just ask, what was the biggest surprise as you were going through this process? As we know, there's all these twists and turns, and just what surprised you about anything in this research? What was that unexpected?
The phenotype for sure. And I also, at the same time, while I was isolating the mutants, I also tried RNA interference. And with that was a quicker method, but obviously RNA, especially with such a large family, you can have multiple off targets. So when I did the RNA, I saw a 50% reduction in nodulation, but it was not at all what we saw in the triple mutant. So it just, for me, it showed that that RNAi can be so, we need several different ways of proving the same phenotype to be absolutely sure what we are seeing is correct. So yeah, reproducibility was what I learned, that this is so crucial for science. And yeah, I'll stop there.
Makes me wonder whether there's some kind of environmental factor that's, the reason that sometimes you see it as in the triple mutant and sometimes you don't. Of course, it could be slight differences in developmental stage, but I do also wonder whether there's some abiotic factor, as much as we try to keep everything the same, that's making
I
this
don't know
difference.
either. I could just speculate on this a bit. For example, even with the auxin mutant, which is the auxin influx transporter that I characterized again as during my PhD, with that OX1 transporter also, if you inoculate it just when the seedling, one day post germination, you don't see the 50% reduction in nodulation. So you have to allow the plants to grow for 1 week, and then you inoculate it. You see the phenotype. So this, how we explained it, was because the auxin transporter shoot to root, and the leaves, the cotyledons have to fully open up for this to then start causing a defect in the nodulation. And this, if so, our hypothesis always was the AMNs are transporting auxin. And if that is the case, is this what we were also seeing? You know, once the plant has to fully grow, and then we see a difference.
Yeah, interesting hypothesis. So we're having some questions coming into the Q&A. Just a reminder to please put your questions there. Anindya Kundu asks, says, there's a higher expression of AMN. Yes, I was worried. I was curious about this too. Higher expression of AMN in NIN1 mutants compared to wild type possibly indicates a negative reaction, right? Negative regulation by NIN rather than its independence. What do you think?
That is possible. Yeah, definitely. But when we say, yeah, that is definitely possible, because if you look at the root hairs in NIN infection, they respond more to, they are hypercurled, and so it's possible that they are negatively regulated by NIN, yes.
Yeah, so do you have any ideas about possible, what could be? Is there something downstream of NIN, right? Is it a stepwise thing, something downstream of NIN that might be actually carrying out this negative regulation?
Yeah, I would just be speculating, and I don't have anything concrete to offer.
Yeah, interesting. Next step. Okay, Marcel Boucher says, which heterologous system would you use to study the transport activity of your ABCB proteins?
I did start this, and I did this in, what we did was in protoplasts, I think, and we tried to we tried this in frog oocytes. That's right. So we took this, the transcript, expressed them in, with Tony Miller's lab, we expressed them in the oocytes, and I used radio-labeled auxin. And we measured how much of auxin was retained in these frog oocytes after we washed off the excess on the outside. But it was on the last day I was leaving John Innes, and it was with a different transporter that I established the protocol, but I could not do it with the AMNs themselves.
It's a good question. So do we have any more? Not at the moment. So one of the things I'm always wondering is this level of overlap and redundancy as well. So do you think these are fully redundant, or do you think the fact that I guess, where am I going with this? The fact that the triple mutant has a mild phenotype makes me wonder what other things are out there that might be redundant with your AMNs. And based on the expression patterns you saw when you looked at all the ABC transporters, are there any possible candidates that jump out?
Yeah, so when I started, again, the genome had just been released, and over time, it just kept improving. So we did find 2 other ABCB transporters, which are also induced in root hair cells, but the expression was nowhere close to the AMNs, but they are still there. And they are also common, they are also expressed during AM symbiosis. So because the ABC transporter is such a huge family, we could definitely, we can have others which can take over the function, even if they are not only responsive to symbionts, right? If they just have a background, low level expression, they could be taking over the function.
And of course, some of the NPF transporters also transport, like NRT 1.1 and Arabidopsis also transport auxin, which gives you a very, very large playing field to look at. Okay, Stefano Leidy asks, do you think an overexpressing line would give interesting insights on their function or role?
I tried overexpressing them, but because when you overexpress the transporter, you don't necessarily overexpress the substrate. So I did not see a phenotype with it. And yeah.
That's a good point. You need the other pieces to that pathway as well. Interesting. So Kaustav Gangopadhyay Padier says, can you shed light on how the three ABC transporters are regulated and what are the structural differences in these three ABC transporters? While the three ABC transporters are of a similar family, how do they function differently?
Okay, so what is differential? Well, that's a really interesting question. I did not find the three to be well, they had very similar expression patterns, but the strength of the promoters was very clearly different. So the AMNs, AMN1 was much weaker in the it needed staining times. I had to prolong the staining times to see the Gus expression during nodulation, and but was very strong in during. No, sorry, it was strong during nodulation, but weaker in microrization. But AMN3, on the other hand, it was so strong. AMN3 was induced in root hairs and in microrized root tissue very strongly. So there must be the other factors which are regulating the levels of how strongly these genes are responding to the symbionts. And that's a really interesting question, but I think more work is needed to answer that.
I think it'll be really interesting to see where they're localized within the cell too, because you may see some differences between them.
Well, yeah,
in
if
that
it's
way.
just in the infection
Yeah.
structures or the yeah,
Or on the outside, yeah, a lot of possibilities. Kevin Cope asks, in a transcriptomics study that I conducted with Medicago, expression of AMN2 and AMN3 was higher in plants colonized by high benefit species of arbuscular mycorrhizal fungi and lower in plants colonized by low benefit species, even though colonization levels and arbuscule densities were comparable. Could you speculate why?
Wow, that is such an interesting observation.
It's really interesting. Yeah.
Well, we should tell me before, Gavin. But so okay, so they are high beneficial and low. So that would suggest something to do with the direct nutrient exchange rather than formation of the structures themselves. That's very interesting. Well, ABCBs haven't been shown to transport any nutrients, but again, they could be transporting. They're not restricted to anything, right? Yeah, that's interesting. Good follow that up.
Very interesting. And of course, with some of the transporters like NRT 1.1, which I mentioned before in Arabidopsis, it was studied for years and years as a nitrate transporter. And then suddenly a study came out saying, oh, it's actually an excellent auxin transporter. So, sometimes we get surprises like that. So it will be interesting to see if perhaps there is an alternative substrate for the ABC transporters. But also just that those AMNs, if they're higher in a high benefit species of arbuscular mycorrhizal fungi, so this means fungal symbiosis where more is transferred, more nutrients are transferred to the plant, that's making it more benefit to the plant. Maybe they're somehow involved in, I don't know, making or stabilizing the infection so that they're more possibilities, more areas, more surface area for transfer. So it's could be indirect that way.
So what do we know about, what do we exactly mean by high beneficial? Is it only in terms of nutrient transport, or is it also colonization intensity? What do we mean by high and low benefits? And we can get.
Yeah, and I think if high benefit is ones that transfer more nutrients to the plant, right? That could be differences at the level of the arbuscule functioning, or maybe number of folds, something like that, how highly convoluted that is.
Yeah, maybe even if we could find phenotypic differences if we tried Kevin's strains with high and low benefit.
Yeah, that's a really interesting question. Many possibilities out there. Okay, well, I think we don't have any more questions, so we will stop there. And please thank Sonali Roy again for a very interesting and informative talk. I've been seeing lots of nice comments showing up in the chat, Sonali, saying how much people have enjoyed the talk. So, and if you would like to learn more, please take a look at her paper, where she has a lot of deep detail and wonderful supplemental figures as well that can bring this all in. And I hope you will join us next month, exactly a month from today, when we have Li Jun Ma, who is our next speaker, talking about what is different between a pathogenic and an endophytic fungus of the same species. So looking at this at a chromosomal level and comparing comparative genomics paired with transcriptomics. So very cool stuff. So we'll be switching gears for next time, but please thank Sonali, and I hope that you will look at and engage with her paper.
Yeah, thanks, Jean, again, and thank you everybody for joining me today and for the support. That was awesome. Yeah. Okay, bye.